Do you find you still get covered in bites during the summer even when you have covered yourself in mosquito repellent? Scientists may have figured out why. The bloodthirsty pests have evolved the ability to sniff out human body odor in more ways than one. Most animals smell with the olfactory receptors in their nose or antennae, which each detect a single, unique scent particle. The receptors are connected to olfactory neurons that transmit information about that particular odor to the brain. However, researchers from Rockefeller University in New York, USA, have discovered that the neurons in mosquito antennae are connected to multiple types of receptor. This means that their neurons are activated by more than one chemical produced by humans, so if one type of receptor is knocked out, they can still track us down. Professor Leslie Vosshall, a senior author of the study, said, "You need to work harder to break mosquitoes because getting rid of a single receptor has no effect. Any future attempts to control mosquitoes by repellents or anything else has to take into account how unbreakable their attraction is to us." Mosquitoes have smell receptors in their antennae that are connected to neurons which transmit scent information to their brain. Each receptor detects a specific smell particle, which triggers the neuron it is linked to. In most animals, each neuron has receptors that detect the same scent particle. However, the New York-based researchers have discovered that mosquito neurons have receptors that detect different particles. This means losing one or more receptors does not affect the ability of mosquitoes to pick up on human smells. Lead author Professor Meg Younger, from Boston University, said, "This project really started unexpectedly when we were looking at how human odor was encoded in the mosquito brain. Mosquitoes are able to track down humans by the carbon dioxide we exhale. And the chemicals in our body odor, including 1-octen-3-ol and amines." Professor Younger's team initially used the gene editing technology CRISPR on female mosquitoes, Aedes aegypti, to deactivate groups of human odor receptors on their antennae. They expected this would entirely prevent their olfactory neurons from firing up in response to the human scent. However, when they measured neuronal activity as the mosquitoes were exposed to human odor, they found the insects could still detect the smell. The researchers then used RNA sequencing to discover what was happening at a cellular level, and found that the neurons that are stimulated by 1-octen-3-ol are also stimulated by amines. Therefore, the cocktail of chemicals in human odor was still managing to activate olfactory neurons through receptors that had not been deactivated. This may be a general strategy for insects that depend heavily on their sense of smell, says Vosshall.